Welcome to week 21, AP Biology students. And in this office hour, we will discuss the gene technology and specifically the electrophoresis, as well as the bacterial transformation with a special plasmid. So about the electrophoresis. For the electrophoresis, we try to sort out the DNA, what we extracted, for example, a blood drop, and definitely the DNA has to be amplified, so it will be the PCR. We will not discuss about that much. And later on, we try to sort out the DNA based on its size. So for electrophoresis, we used an agar blood, and we have channels. Here is 1, 2, 3, and 4 at this point. These are going to be the wells. Well, we place the DNA, a solution with the DNA in it, and we try to make them run the gel. Let's say we have people in here. This is X and Y person. This is going to be a female organism. This is a male. This will be a scenario, and this is the Z as the child. So at this point, we want to just see whether the child relate to the mom and to the female and the male organism, and this will be the latter. Shows the actual segments, DNA segments, what we have in the mixture. So for that, we will have, here is 1, based on the size of the DNA will run a certain distance from the well towards the other side. Why is that important? I mean, how can we manage that running? The well area is going to be positive. We put a solution in the chamber, the electrophoresis chamber. Oops, this will be negative and this will be positive because the DNA is a negative macromolecule polymer. So it want to just move towards the positive sign. Now, the DNA, based on the size, will be able to move towards the positive, and the more distance the DNA was able to make, the smaller the DNA is. So these are smaller DNAs. Think about that the agarose gel is a obstacle course, so the DNA has to run through. The smaller it is, it can go through easily, and the longer the DNA is, it is harder in the obstacle course for that. This is a larger DNA. So what we have in here is, for example, the X person's DNA will run this way and that way. The Y person's DNA runs here and here only, okay? And the Z person's DNA will be here and there. We can just have this one in here. Good. So what can we conclude from that electrophoresis? That the Z person shares same length DNA in the repeated sequences, in its non-coding repeated sequences in the DNA with person X, but Z does not have any DNA sequence, similar non-coding repeated sequences with Y. So we can conclude there's no blood relationship between Z and Y, but there is between Z and X. More short segment DNA we check, the more we can really relate people to each other. So that is about the gel electrophoresis. And we go and discuss the plasmid transformation, bacterial transformation. Before that, I want to just point out that we talked about operon, and it is really costly for a cell to produce proteins. It is an anabolic reaction, and if the cell doesn't need that protein, such an enzyme, why to make that protein? Why to deal with the transcription as well as translation? This is super important on that. So we talked about the bacterial operons, and operons are a promoter and many genes downstream. And between the promoter and the genes, there's an operator segment where an operator will just get over there to let the transcription not to move on. So this was the tryptophan and the lac operon. So the lac operon was an inducible operon. Lactose in the cell or around the cell started to induce that operon to do the transcription and later on translation. Now, here is a bacteria. The bacteria has a genetic material, and it can pick up a plasmid from its surroundings. If it's picking it up, it's called transformation. The transformation, so in the surroundings or from dead bacteria, the plasmid that codes for not essential proteins, but some additional proteins like enzymes that break down antibiotics. So the antibiotic will not going to kill the bacteria. They can be on the plasmid. So that plasmid can be picked up, heating up the bacteria, cooling it down pretty fast. They usually pick up a plasmid. Now, this is transformation. We have a plasmid in here. I will make it a bigger one, and it's a circular, and it contains an origin or I region. This is usually where the DNA polymerase gets on and makes copies of this plasmid. Now, this plasmid can stay in the bacteria, or they can form pili. Let's say this plasmid is in here, and they form pili in here with another bacteria that does not contain the plasmid. So it's a conjugation when this replicated plasmid is going to go through into that bacteria that does not have a plasmid. So it's called the conjugation. Good. So now it will have that. Think about that antibiotic resistance is on that plasmid. So this bacteria that was not resistant to antibiotics, so ampicillin, streptomycin, penicillin would have killed that bacteria. Now, this bacteria is not going to be killed because it has a resistance on that. It can break down the antibiotics around it. So this is the ORI site for that. Now, for us, there will be a segment on this DNA that is the ERA C. The arabinose C, it's going to make a regulatory protein. And the regulatory protein starts with a capital ERA C. This is the protein. And it will go, this is a promoter region. It's very similar to the operon. And we have a region in here that was a list of a serial of genes after the promoter that was able to break down arabinose. Arabinose is a sugar, very similar to the lac operon, inducible operon. But at this point, engineers, cut this segment out. Actually, let's put this one in here because this will be the operator segment where that ERA C was able to combine to stop the RNA polymerase to read and transcribe the enzymes that they are important for breaking down the arabinose. But now they changed that segment and they took out this gene and they added a gene, the green fluorescent protein. So when arabinose was present, this regulatory protein got off from the operator site, and the RNA polymerase was able to copy that gene to make a green fluorescent protein that came from a jellyfish. This one also contains another segment. This is the EMP. This is producing an enzyme that breaks down the ampicillin that would kill the bacteria. So it's relates to the antibiotic resistance in here. This is having a different promoter section, so it does not depend on that promoter, that operator segment in here. So what this one is going to tell us, if we have a bacteria, this is a bacteria, without the plasmid, if I put that bacteria on a nutrient agar, all the bacteria will be able to spread all around. But if I put this bacteria on an agar with ampicillin, this is antibiotic, there will be no bacteria going to grow on that agar. So it will be totally clear because the ampicillin will kill that bacteria that does not have any antibiotic resistance. On the other hand, if I get this bacteria with, how about I use the same color? This is the bacteria with the plasmid. If I put that bacteria on that plate, we will be able to see, so not in here anymore, we will see colonies all around. They will be able to live in here because they can break down the ampicillin. And if I would add ampicillin, actually arabinose to this agar plus agar for the bacteria. In this case, bacteria who was able to pick up that plasmid would be present on here. Even they would glow green, they would form green colonies all around because the arabinose start to turn this operon on, the RAC will get off, the RNA polymerase will be able to translate the GFP gene that is going to stand for green fluorescent protein that is going to be produced. Why is that good for the bacteria? Well, it does not good, just to glow, but this is how engineers started to modify this plasmid. And now with this protein, actually, it's super good, this segment, because if they add this gene to any other DNA segment that produces protein, the protein will start to glow green, so they can trace its location in the cells. So it's super interesting what technology can, the gene technology can do, biotechnology. So the bacteria on the arabinose, if I would have this bacteria without the plasmid on the arabinose agar, we would have bacterial colonies all around too. Okay, so it would. But if I add ampicillin or some antibiotics in here, and if the plasmid does not have antibiotic resistance gene that codes for an enzyme to break down the antibiotics, that bacteria would not be able to live on an agar that contains that specific antibiotic, an enzyme that breaks down the antibiotics. Okay, I think that's all basically for you today.